LSZ102, you see the structure here. It's a benzodiazepine system, a carboxylic acid, and a fluoroaromatic. The early phase synthesis started from 3-methoxythiophenol. Then the first step was alkylation of this bromoacetaldehyde diethylacetal in acetone, 90% yield. Then with boron BF3 diethylacerate in dichloromethane, only very low yielding. This intramolecular Friedel-Crafts acylation. Then with metachlorobenzoic acid, the benzodiazepine system was oxidized to the sulfone, followed by bromination in dichloromethane, and then reaction with 4-bromophenol on cesium carbonate as a base in this SNAR reaction. Then the next step was sodium borohydride reduction.
Excuse me, would it be possible to have full screen, please? Because your molecules are very small and it's quite difficult to see.
Is it better?
To put your, yes, it's much better.
Oh, sorry. Okay.
Thank you. Thank you.
Okay. Then the next step was the reduction of the bromine in 2 position with a sodium borohydride, followed by dival reduction of the sulfone to the benzodiazepine system. Then the next step was Heck reaction to acrylic methyl acrylate, followed by bromination again, ester hydrolysis. So very long sequence actually with a lot of oxidations, reductions. And in the next step, you see the upcoming step that they were run on the GMP. Then afterwards, there was the first step was the debromination with PBr3. This had to be purified by column chromatography. And then there came a Suzuki coupling, palladium catalyzed Suzuki coupling. The LSZ102, actually the drug substance crude product, could be isolated as the dicyclohexylamine salt, which then was liberated again as the free acid and purified by column chromatography. We had 2 major byproducts, so this vinylic impurity with HF elimination, actually, and the des-bromo product of the starting material, just the des-bromo, which is a usual byproduct in a Suzuki coupling. And this one could be removed with the RP chromatography. The other one stayed in, but that was tox qualified up to 0.6%. The isolation of the final product was then done just by a pH setting. So first it was dissolved in sodium hydroxide, so as a dianion, and then after addition of HCl, the product precipitated out as a amorphous solid. There were still some issues because we had TBME in the system, and there was TBME incorporated in the drug substance, and also the sodium chloride, which was formed with this pH setting, was also difficult to remove because the amorphous solids entrapped sodium chloride. The side chain here, we started here from this bromo aromatic, was in a Miura coupling under with boron pinacolato, palladium bisphenylphosphine dichloride, potassium acetate, and then this was actually purified by column chromatography on early stage. The first improved early phase synthesis was then the dicyclohexylamine salt could be purified with 2 recrystallizations from methanol water, actually including a charcoal treatment to remove last traces of palladium. And then the isolation of the final product was done in the same way. So first dissolved in aqueous sodium hydroxide, extraction of the dicyclohexylamine with heptane, followed by acidification and then precipitation of the drug substance. Then the early phase non-GMP steps were improved. So we started then we made use of the HIGA cyclization, which starts from methoxy 4-methoxycinnamic acid and thionyl chloride, gave this adduct 19, and this was then trapped with methanol to give the methyl ester 20 here in over the 2 steps in 47% yield. The carboxylic acid in 2 position or after ester hydrolysis under sodium hydroxide DMSO at elevated temperature gave the decarboxylated product 21. This was then oxidized to the sulfone, and the reaction with 4-bromophenol yielded the intermediate 8, which was then the same common intermediate as in the other synthesis. And then the reaction continued like the same way. So with diol, the sulfone was reduced to the benzothiazine, then Hecker relation, NBS bromination, ester hydrolysis, and then we saw on intermediate 13 actually a byproduct, a 7-chloro byproduct, which was co-eluting in the earlier steps under HPLC. This was a carryover of the first step, so the Higa cyclization of 4-methoxycinnamic acid with ethynyl chloride, and that had to remove in an upcoming step after BBR3 demethylation by twice recrystallization. Then finally, the improved final early phase synthesis started from the same system, but we then, from the compound 21, instead of oxidizing to the sulfone, we oxidized only to the sulfoxide. That was possible to do that in 69% yield with hydrogen peroxide in the presence of acidic acid and in dichloromethane, and then we could actually do the SNAR reaction with 4-hydroxycinnamic acid methyl ester, and then the sulfoxide could be reduced to the benzothiazine with DMS chloride triphenylphosphine. These are compatible reagents to the ester moiety, whereas the diol for the reduction of the sulfone was not compatible with an ester. So this was an improvement by 2 steps, and then the next steps were on the same sequence. However, BBR3 was still required in this early phase synthesis, and BBR3 is a nasty reagent, especially on large scale. It's difficult actually to ship. It's also difficult to handle because it's very corrosive, and it's not easy to handle reagent for large scale. The side chain synthesis actually started as I described before. So with this bromoaromatic system 18 with bisphenol pinacolato diboron, palladium in a Miura coupling with palladium bistrifosine dichloride, potassium acetate. And then it was purified finally by a low temperature crystallization. It has a melting point of only 33 degrees. We could crystallize it from heptane at minus 50 degrees in a yield of about 64 to 68%. However, there was another problem. Besides the low melting point of only 33 degrees, it exhibited a strong thermal decomposition energy in the DSC. And we had distillation processes, so from dioxane to heptane, and that was on scale exhibited thermal safety problem for large scale. So that's why we actually wanted then to get away from that. The synthesis of this compound 18 is described here. So it started from 2-bromo 5-fluorobenzoic acid with ethynyl chloride. The acid was converted to the acid chloride, then chain elongation, and after with diethyl malonate, magnesium chloride triethylamine, followed by decarboxylation of the beta-keto esters to the acetophenone. There were these 3 steps in pretty high yielding, 91%, and then with SF4HF, the acetophenone was transferred to the difluoro compound. So the non-GMP part, the oxidation to the sulfoxide, that solved the problem with the bromodioxine issue, because the bromodioxine issue was the following: 4-bromophenol, the purchased 4-bromophenol contained polybrominated phenols, and they potentially can form bromodioxines, which are very toxic. And with this actually final early phase synthesis, we could avoid the use of 4-bromophenol. We used the 4-hydroxycinnamic cinnamic acid methyl ester. And the 7-chloro impurity, which I showed you a few slides before, that was under control due to process improvements on the first step. So the challenges of this early phase synthesis was still long linear sequence, the use of PBr3 on large scale, the use of this boronate ester 17B with a low melting point and a high thermal decomposition energy, and this triggered the need of an alternative synthesis for final process. We thought actually, why not using a halo cinnamic acid, and the halo atom would be the masking of a hydroxy group, which is shown here. The process actually made the same sequence, so the Higa cyclization to the methyl ester, the yield could slightly be improved to 55% yield, decarboxylation followed by oxidation to the sulfoxide, again the SNAR with 4-hydroxycinnamic acid methyl ester, followed by the sulfoxide reduction, and then ester hydrolysis here to the bromo-cinnamic acid. And then we made use of the Buchwald reaction. So the conversion of a bromo aromatic to the phenol under a palladium catalysis with tertiary butyl X-phos and potassium hydroxide. And from this compound 33, which was a known impurity actually coming from the Suzuki coupling, we could go both ways. We could brominate to the bromo, 2-bromo benzothiazine system and then go via the Suzuki route. Or we thought, why not going directly into a CH activation with the precursor of 17B, so with this 18 under palladium catalysis, and then solve the issue of this boronate 17B with a low melting point and this thermal decomposition energy. The development of the aryl bromide to the phenol, that is shown here. So we used only 0.3 mol% of this palladium allyl chloride dimer and 1 mol% of the tertiary butyl X-phos. According to this Buchwald procedure, at the beginning, 3 equivalents of potassium hydroxide were added. After about 3 hours, there was 17% conversion achieved. Then another 1 equivalent was added, potassium hydroxide. And after stirring overnight at 60 to 65 degrees, a complete conversion was achieved. In the starting material, 32, the bromo derivative, we also had the chloro impurity. This was also reacting to the desired product, but only when all the bromo compound was consumed. So this reaction was actually executed on 4 batches on 85 and 100 kilo scale. And after the campaign, it was found in the lab that actually the phase transfer catalyst tetrabutylammonium bromide was not actually required. The palladium was, which was the residual palladium, was removed with sodium dimethyl dithiocarbonate wash. Although this, there is a slight danger of forming a carbon CS2 after acidification of the aqueous phase. So this had to be carefully investigated before going to the pilot plant. The next step, the CH activation. We found conditions, the palladium acetate and cyclohexyl diphenylphosphine as a ligand. The catalyst was in the first scale-up batch preformed with 4 mol% palladium acetate, 8 mol% of cyclohexyl diphenylphosphine in THF, 5 equivalents of sodium tertiary butoxide solution in THF, which is commercially available, were added. The reaction was heated up to 65 degrees for at least 14 hours, and usually we had greater than 98% conversion. The reaction is very water and oxygen sensitive. After complete reaction, the THF was partially distilled off. There was an aqueous workup done. We added 2-methyl THF, and then the dicyclohexylamine salt was actually isolated again, usually with around 97% purity. Then this was the dicyclohexylamine salt of the drug substance was dissolved in sodium hydroxide and extracted with heptane. So the dicycloxylamine was removed in the heptane phase, and then the monosodium salt of the LSZ was crystallized at pH 7.8 by addition of phosphoric acid and N-acetylcysteine to remove last traces of palladium, and the product could be isolated in about 70 to 75% yield. And the purity was usually about 99.3, and the palladium levels were less than 5 ppm. And there were 3 batches executed on 40 to 50 kilogram scale. We also found that the palladium acetate is, the quality of the palladium acetate is critical. There is literature also of authors from Johnson Matthey. Colacott here is an example. He made a study of actually palladium acetate, and he recommended actually to run NMR of palladium acetate in dichloromethane. And here on the left-hand side, you see palladium acetate, which was not suitable for this reaction. In the methyl region, you see a lot of peaks, whereas this one, that's a good batch of palladium acetate. You see mainly only one signal here at about 2 ppm of the methyl group of the acetate. In the first scale-up batch, we could actually isolate N-acetylcysteine adduct, which was coeluting with the starting material, but we only found out when we took the UV spectra of the of this byproduct, and it was found after HR-LCMS that it's a N-acetylcysteine adduct on the drug substance, and the N-acetylcysteine wasn't washed out very well because the product filtered very badly in the pilot plant, and then after drying, it just gave this microadduct. We also could isolate and identify 6 impurities coming from the CH activations, which were shown here in this slide. So the isolation strategy of the API was also ongoing. Up to this point, we isolated the drug substance as an amorphous solid. In the first phase 1 batches, it was coming from a precipitation process. And later on, we did spray drying. However, on scale, the spray drying was very low. The yield was very low. It was less than 50% due to plasticization of the product in the cyclone. Therefore, a crystalline API was intensively sought. And finally, our colleagues from our particle engineers found a final crystalline form of the free acid and also a crystalline co-crystal of LSZ with beta-ine. And the new isolation strategy, because we had this with the monosodium salt, we had a very bad filtration and very long filtration time, was as follows. So still doing the CH activation of this compound 33 with the bromoaromatic palladium acetate, the same condition with sodium tetrabutoxide. Then doing first dicycloxylamine salt formation. This filtered very well, removed a lot of impurities. Then the dicycloxylamine was extracted from aqueous phase. Then back extraction of the LSZ into the organic phase, and then we did a beta-ine co-crystallization in acetone. This removed all these byproducts, which I showed in the previous slides. And then with in acetic acid water, we could actually crystallize the free acid as a crystalline solid, and the beta-ine was removed into the mother liquor. And here were the results of the beta-ine co-crystals of 4 production batches. So on 45 to 55 kilos, the yield was between 75 and 85%, purity above 99%, palladium was below 5 ppm, and the dicycloxylamine, which was Ames positive, was also below 5 ppm. We then turned our attention again to the non-GMP part, because as you might remember, we had this oxidation reduction sequence. So we had oxidation of the benzothiazine system to the sulfoxide, or previous to the sulfone, then did the SNAR reaction, and then reduction of the sulfoxide or sulfone again. So we thought, I mean, in the Higa cyclization, we are actually doing in the 2 position, we are ending up with an ester. And the ester is also an electron withdrawing group. We thought, well, maybe we could try the SNR reaction with the compound 37. And first, the initial trials were done with the tertiary-butyl ester, because this is not prone to hydrolysis under basic conditions. However, the tertiary-butyl ester was not stable under reaction conditions. However, we found that the ethyl ester seemed to work. It gave about 50 to 60% conversion under strictly anhydrous condition using potassium carbonate and tetrabutylammonium bromide in DMSO. Then later on, the esters, 2 esters were hydrolyzed, or they're partially already hydrolyzed during the reaction conditions of the SNR reaction. And then afterwards, the decarboxylation was possible with potassium carbonate in DMSO, or even the milder base potassium acetate in DMSO at 110 degrees. Overall, the yield of the sequence 37 to 32 was around 20 to 25% on kiloscale on the new route. The old route was slightly higher, however, 2 steps more, which were a bit HSE unfriendly. So the oxidation to the sulfoxide with hydrogen peroxide in dichloromethane in the presence of terafluoroacetic acid, and then followed by the reduction of the sulfoxide with terafluorophosphine and TMS chloride, which gave terafluorophosphine oxide, also waste. So obviously this process requires further optimization. Then our proposed final synthesis is as follows. So starting from 4-bromocyanamic acid in the Higa cyclization, and then ethanol to make the ethyl ester 37, then doing the SNR reaction, followed by the hydrolysis to give the compound 39, and then the decarboxylation of the 2-carboxylic acid of the benzaldehyde system, conversion of the bromo to the hydroxy, CH activation, and finally isolation of the beta and co-crystal, and finally making the free LSZ102 as the carboxylic acid in a crystalline form. In a nutshell, the project achievements in 4 years. So the synthesis from a 10 non-GMP and 8 GMP steps. The phase 1 steps could be reduced to overall 7 steps. We could avoid critical steps, reactions like PBr3 for bromophenol, and this intermediate boronate ester with a low temperature melting point, low temperature crystallization, and thermal safety issues. 6 different synthetic schemes and 7 different campaigns, and the first implementation of CH activation in our company on 50 kilogram scale. The catalyst going from air-sensitive tetrakis-driven phosphine palladium to a stable palladium acetate plus ligand, and the drug substance solid from an amorphous to crystalline. And this work could actually be published in the Journal for Organic Process Research and Development in 2020. I would like actually to thank a lot of people in our company in China, in our colleagues from China, as well as here in Basel, Switzerland. A lot of people from external companies who made different campaigns or supporting activities. And I would like to thank you for your kind attention.